This year's Nobel Prize is awarded for determining how oxygen levels are sensed by cells. Oxygen is essential for life and is used by virtually all animal cells in order to convert food to usable energy. However, the amount of oxygen available to cells, tissues, and animals themselves can vary greatly. This prize is for 3 physician scientists who found the molecular switch that regulates how our cells adapt when oxygen levels drop. The most fundamental use of oxygen by the cell is to convert food to usable energy. Just as a candle needs the right amount of oxygen to burn cleanly, cells need to adjust their metabolic rates based on how much oxygen they have available to them. This allows each cell, and indeed our bodies, to efficiently and safely burn fuel so as to create heat, do work, and build new tissues. Cells and tissues are constantly experiencing changes in oxygen availability. As an embryo grows and develops, as muscles work, the oxygen available changes as the tissues themselves change. Cells need a way to adjust to the amount of oxygen they have while still doing their important jobs. Sometimes, oxygen levels change across the entire body, such as when we go to high altitudes, and sometimes they change in very small parts of our bodies, such as when we get a wound that interrupts the local blood supply. This triggers an adaptive process called the hypoxic response, which in turn can induce processes in the body as diverse as new blood vessel formation, or angiogenesis, new red blood cell formation, or erythropoiesis, and metabolic adaptations of cells, including glycolysis. One of the first physiological changes caused by low oxygen noted by scientists was the erythropoietic response. This occurs when the body is at high altitude or when it experiences anemia, such as an insufficient number of red blood cells. This hypoxia, or low oxygen state, induces expression of a hormone by the kidney, the erythropoietin hormone, also known as EPO. It became clear to researchers in the field that EPO gene expression goes up when oxygen levels go down. This allowed scientists to use the EPO gene as a tool to analyze the role of oxygen in the control of gene expression. Now we turn to the work that gave rise to this year's Nobel Prize. The earliest work by one of this year's laureates, Gregg Semenza, showed that this increase in EPO was due to a specific region of the EPO gene that he defined and called the hypoxia response element, or HRE. Semenza used this oxygen-regulated segment of the gene to isolate a transcription factor protein complex. He called this protein complex HIF, for hypoxia-inducible transcription factor. HIF itself is formed of 2 proteins, one called ARNT, or ARNT, that is always present, and one he called HIF-1 alpha. The HIF-1 alpha protein is oxygen sensitive, which is to say that it is present when oxygen is low and disappears when oxygen levels are high. Semenza and others showed that HIF was present in almost all cells, and at this point HIF was potentially a key regulator of oxygen responsiveness. Further work by a number of groups showed that HIF was constantly made by cells, but in high oxygen conditions, which we call normoxia, it was constantly destroyed by proteasomal degradation. At the time of the cloning of HIF by Semenza in 1995, William Kaelin was studying a gene involved in cancer called the von Hippel-Lindau gene, or VHL. His group, in collaboration with others, showed that cells lacking VHL had increased expression of hypoxia-inducible genes. Subsequently, Sir Peter Ratcliffe's group showed that VHL was necessary for HIF to be destroyed by the proteasome. This indicated that a key role for the VHL gene was to direct HIF to be destroyed under high oxygen conditions. However, we still didn't know how oxygen regulated this switch. Ratcliffe and Kaelin then simultaneously showed the way by demonstrating that an oxygen-dependent reaction seals HIF's fate in high oxygen conditions. When enough oxygen is around, two prolines in the HIF-1 alpha protein are hydroxylated. Hydroxylated HIF is then recognized by the VHL complex, which then targets HIF for destruction. To sum up, then, this elegant switch, which was elucidated by the work of this year's laureates, under low oxygen conditions, HIF is made, it accumulates in the nucleus, and that results in the induction of a wide variety of oxygen-sensitive genes. But when oxygen is present, HIF is hydroxylated, recognized by VHL, and destroyed. There are at least 300 genes with hypoxia response elements, or HREs, in the human genome. These are all regulated by HIF to help cells, tissues, and the entire body cope with changes in oxygen levels. Some of these genes include those encoding the vascular endothelial growth factor, or VEGF, that regulates angiogenesis, erythropoietin, and almost all of the enzymes involved in glycolysis, or anaerobic fermentation. The effects of HIF range across almost every aspect of physiology, from metabolism to exercise, to embryogenesis, to immunity, and the changes wrought by altitude. Diseases where HIF response plays a critical role include anemia, cancer, stroke, infection, and heart attack. Applications of these findings are already beginning to make their way to the clinic, with potential drugs to increase HIF used to treat anemia and to suppress HIF used to treat some forms of cancer. These fundamental findings have greatly increased our understanding of how the body adapts to change, and applications of these findings are already beginning to affect the way medicine is practiced. This year's 3 laureates have greatly expanded our knowledge of how physiological response makes life possible.